Okay, good morning everyone. So this is a new video on the light dependent stage of photosynthesis. We'll also talk a little bit about the light independent stage, but the main focus will be the light dependent stage. Okay, we're going to start off with a few recaps from the last videos. So, what do we know so far? We know that photosynthesis takes place in chloroplasts, and more precisely in thylakoids. So these oval-shaped compartments, they're the thylakoids, which are in chloroplasts. And you know that the little green dots, they are photosystems, and we know that there are different pigments. We know they play a role in photosynthesis thanks to absorption and action spectrums, and we started talking about this idea that some of the pigments, so a pigment chlorophyll A in fact, can transfer electrons. So we'll see how that comes into play. So that's one first big idea. There are pigments in the thylakoids and they're going to transfer electrons using the energy brought by light. Okay, the second thing we know, we studied Rubin and Cameron experiments, and we know that the oxygen which is produced throughout photosynthesis comes from water. Okay, and we deduced from that photosynthesis is two things. It's the oxidation of water, and it's coupled with the reduction of carbon dioxide. So as I said before, photosynthesis can be summed up by these two things: oxidation of water and reduction of carbon dioxide. So what we'll see is that the oxidation of water is part of the light dependent stage. This means that you need light. Light is needed to break water into dioxygen, 4 hydrogens and 4 electrons. And the second part, the reduction of carbon dioxide, doesn't need light. So it's part of the light independent stage. Okay, so that's for the first recap. So we'll see how all these can be put together so that you have the final picture with all you've got the pieces of the puzzle, but we simply have to put them all nicely to see the global picture. Okay, I'm going to ask you to move to this diagram. So we're going to label a few things and explain a few mechanisms which are happening in this diagram. So let's set this diagram and see where we are to start off with. So we're looking at a granule thylakoid. So one of the thylakoids which is part of a granum. So number one is going to be simply the interior of a granule thylakoid, the inside. Number two is going to be the granule thylakoid membrane. The membrane will be similar to the membrane of the cell membrane, so made up of phospholipids, etc. And number three, so this liquid inside a chloroplast is called the stroma. So we've already labelled this on the main diagram, but it's important to just recap where we are. And I would advise you maybe to have a look at your general diagram of the chloroplast, just to really make sure you know where you are within the chloroplast. Okay, so last thing, this is new for you. ATPase complex. So it's a complex. We'll see that we could say produce ATP. Perhaps the better word would be regenerate ATP, but you can also say produce ATP. And last things, I didn't really want to label them because there are already a few labels and we'll look at it in detail. But so PS2, so it's not PlayStation 2, it's Photosystem 2, and PS1 is not PlayStation 1, it's Photosystem 1. T1, T2, T3, T4, these are proteins. We'll see later on that they carry electrons around, they transport electrons. Okay, a few molecules that we've already talked about: NADPH2, ATP. Okay, I'm going to move on to the next slide just to have the labels incorporated in the diagram. Okay, so we've set up where we are, so we're within the thylakoids, and we're now going to talk about function. What are the different mechanisms which occur within these thylakoids? Okay, so the first thing we have, so this is something you know now, that water is transformed, broken down into oxygen, protons and electrons. So the electrons, they're going to go off into towards different proteins. They're going to go through the photosystem and towards different proteins. And the H plus is protons. They're going to go through the ATPase complex, and the dioxygen is simply going to come out through the membrane by diffusion. So this first step is a process where water is going to be split. We've got the splitting of water. Just a little joke, you might think of Moses, who was splitting the Red Sea, but chloroplasts can't do that just like Moses, because he had a stick and chloroplasts don't have sticks, so they can't do it in the same way. So instead of that, they are going to use light. If you see on the diagram, you have light coming in, so light from the sun, for example, and therefore, because light is used, this splitting of water is going to be called photolysis of water. So we've got two words in photolysis. We've got photo, so photons, light, and lysis, which means to split, to break down. So this is something you need to know. You can red star this. Well, obviously the whole diagram, but you can have back questions where you can be asked to explain what photolysis is. So it's when water is split into half O2, 2 protons and 2 electrons. Okay, so first thing, light comes in, goes through the photosystem pigments, and that will help split water. So this is photolysis of water. Okay, the next thing, we're going to have proteins which transport electrons. So the electrons, which after they've been taken from water, they're going to follow the following path. So go through the photosystem, go through the proteins, go through a second photosystem, and go through another protein. And at the end of the day, these electrons, they're going to arrive in the stroma, so they're here, and we'll see afterwards what's the next step. But so the electrons, they started here, so they come from water, these electrons, and they go through this path. So just to illustrate what it could look like, a bit like the dwarves in the Hobbits, the electrons are thrown around from proteins to proteins, but maybe in a little bit more organised way. So simply, this is the transport of electrons. It's not really an official word, but this is how I've named it. So electrons are transported through different proteins. I'm sorry, I said protons, I meant electrons. So electrons are transported through different proteins. Okay, so third part, our electrons, they are going to be used, added to NADP and with 2 protons. Oh, this is the problem of doing it live. I'm just going to move the phone so that we're not disturbed. Actually, I might be able to, just going to try something new. I'm going to pause it and see if I can come back to this. Okay, so hopefully this worked. I'm just going to reset my pen. Okay, so we're now talking about the regeneration of NADPH2. Whoops. Okay. So we've talked about photolysis of water. We said that just like the dwarfs and plates, proteins are transporting electrons. Okay, so transport of electrons, and last thing, regeneration. So third thing, regeneration of NADPH2. So as I was saying, NADP, 2 H pluses and 2 electrons, and they're going to add up and we'll have NADPH2. So we did this in the last video, so quite similar to the Hill reaction, but in this case this is the actual molecule NADP. Okay, and last thing, so the protons, which also come from the splitting of water, the protons, they're going to go through this ATPase complex, and you can see it here. We've got ADP plus PI, so this PI is inorganic phosphate, is regenerated in ATP. So we're going to detail this, and this is called the regeneration of ATP. So just to sum up, so in the thylakoids and around we have a stroma, we have different things. Light comes in, this provokes the photolysis of water. The electrons are going to go through different proteins and they will be used to regenerate NADPH2. And on the other side, protons are going to go through this ATPase complex and we're going to regenerate ATP. Okay, so we're going to talk a bit more about this number 4 here. Okay, so the ATPase is a set of proteins. There are at least, you can only see 7 on this one, but there are at least 11 or 12 proteins which all help or have a function in this ATPase. So this is part of something called the chemiosmotic theory. So we've got 2 words. We've got osmotic, so you recognize the word osmosis, and we've got chemio for chemistry, chemical. So the idea is that we've got a mechanical force. The fact that the protons go through the ATPase, so following the law of osmosis, so where there is a bigger concentration, so there are more H pluses here, they're going to go down their concentration gradient to where there's less protons, so from high to low. So this is going to create a certain force. These protons going through the proteins of the ATPase is going to provoke some changes in the shape of the proteins, and that's going to create energy. And this energy is going to be used to regenerate this molecule here, so ATP. So you know from the lessons on DNA that we've got the sugar, the nitrogenous base, and different phosphate groups. So when we've got ATP, we've got 1, 2, 3. So ATP is for adenosine triphosphate, Okay, and so when the protons go through, we're going to transform ADP into ATP. So as I said, the protons go through, it changes the shape of the proteins. That's going to create some energy, and this energy is going to be used to add a phosphate group to ADP and create ATP. So in the body, or in plants, well, in many organisms, there's a constant renewing of ATP. So you add phosphate to ADP to create ATP, and then ATP is used somewhere else in the cell. So the phosphate group comes back off, and we're back to ADP. So this occurs a lot, all the time. So we've got this ATP-ADP cycle. So for the moment, just to be clear, in the light-dependent stage, we're doing the first part, we're regenerating ATP. So we're going from ADP to ATP. This is in the light-dependent stage, and we'll see that in the light-independent stage, it's going to be the opposite. ATP is going to be used, and therefore ADP will be recreated. Okay, so that's for chemiosmotic theory. So same thing, you need to notice the fact that we use mechanical energy, so through osmosis of protons, to create a chemical which has energy, and this chemical is ATP. Okay, this is a diagram which recaps quite a few things. I would really advise you to learn this one by heart so that you don't have any problems for what we'll learn next. So, if you read the title of this part, and that's what your syllabus says, you need to know the substrates and the final products. So, the substrates are what we start off with. So in this case, for the light-dependent stage, so the left side of the diagram, so all this part in yellow, we'll look into that. What's the substrate, what's the products? And on the right side we've got the light-independent stage. So we'll detail this in the second part in another video. So substrates, so we're going to start off with, so we're doing these substrates and final products of the light-dependent stage. So we start off with water. Okay, this diagram is balanced so that we have the full equation of photosynthesis. 12 H2O plus 6 CO2 will give 6 O2 plus glucose plus 6 H2O. So if we start off with 12 molecules of water, we're going to have photolysis. I'm going to put PL for photolysis. We're going to end up with 24 electrons, 24 protons. So one of the first products, I'm going to put a P for products, we're going to have 6O2. So this is a substrate. So 12 water is a substrate, that's what we start off with. And one of the products is 6 dioxygens. But we have other products. We're going to have the regeneration of 18 ATPs. Okay. And also we're going to have the regeneration of 12 NADPH2s. So if we sum this up, this is something you need to know. We start off with 12 molecules of water. That's for the light dependent stage, and we're going to end up with 6O2 plus 12 NADPH2 NADPH2, and we're going to end up also with 18 ATP. Okay, so here we go again. So if we do a final recap of this light dependent stage, the substrate is 12H2O, and at the end of the day we're going to have 6 dioxygen, 12 NADPH2 and 18 ATP. So when we say that we use the energy of light in photosynthesis, it's true, and some of the intermediates are going to be NADPH2 and ATP, which are molecules which have energy in them, because if you break off the protons or if you break off the phosphate group, that's going to create energy. Okay, so this is the recap. Very briefly we're going to look at a few things about the light independent stage this time. What we can say is that NADPH2, ATP are going to be used up, so they're going to go back to NADP form and ADP form, and this will be used to reduce CO2. So thanks to NADPH2 and ATP, CO2 will be reduced to glucose, and water will also be produced. So the main missing part is what happens here. Okay, that's something that is called the Calvin cycle. So as I said, this diagram you need to know it, and another diagram just to have a very basic recap. And this really shows that we've got two things. We've got the oxidation of water, which will be transformed into dioxygen. We've got the reduction of CO2 into CH3O and glucose if you've got 6 CO2. On the left side we have the light dependent stage, and in the stroma of chloroplasts we've got the light independent stage, and one of the main things is this Calvin cycle, so we'll look at the detail of this. And last thing is that in the light dependent stage ATP and NADPH2 are produced. They're used up in the Calvin cycle, which will help reduce CO2, and then we're going back to NADP, ADP, which will be regenerated once more in the light dependent stage. So all these are part of different cycles. Okay, so in the next video we're going to detail. Going to go back a bit. So in the last video, in the next video we will detail this part here. So how these electrons are transported throughout the proteins, and after that we will detail the Calvin cycle, what we just saw. Okay, well, sorry for there were a few hazards here and there, but I think we got through it. So have a good day. You'll see that in the homework you can have a look at the back exercise from 2013, so questions 1 and 2, and that will help you understand a little bit more how scientists were able to prove that ATPase does what it does. And that's all for today. Goodbye, have a nice day, and I'm just going to close everything as usual. Bye bye.